Scientists discover a new mechanism that prevents the proliferation of cancer cells. University of Montreal, Canada. Article: Canadian researchers have discovered a new and direct molecular mechanism to stop cancer cells from proliferating. In the prestigious journal Nature Cell Biology, scientists from Université de Montréal show that a disruption of the fine balance in the composition of ribosomes, which are huge molecules that translate the genetic code into proteins, results in a shutdown of cancer cell proliferation, triggering a process called senescence. Quote, ribosomes are complex machines composed of both RNAs and proteins that make all the proteins necessary for cells to grow, end quote, said Université de Montréal biochemistry professor Gerardo Ferbier, the study's senior author. Cancer cells grow and proliferate relentlessly and thus require a massive amount of ribosomes, he explained. Growing cells must coordinate the production of both ribosomal RNAs and ribosomal proteins in order to assemble them together in strict proportion to each other. Ferbier said, "We were surprised, however, to find that if the production of ribosomal RNA protein proportions are driven out of balance in a cancer cell, proliferation can be shut down in a very simple and direct manner." In their research, led by Université de Montréal biochemistry researcher Frédéric Lessard and done in collaboration with biochemistry professor Marlene Offinger of the Université de Montréal affiliated Montreal Clinical Research Institute, Ferbier and his team uncovered a new mechanism that uncouples ribosomal RNA from ribosomal protein synthesis to stop the proliferation of cells bearing oncogenic mutations. The team demonstrated an unbalanced ribosomal RNA and ribosomal protein synthesis during oncogene-induced senescence, a response that prevents cancer formation. In the lab, senescent cells shut down ribosomal RNA synthesis but kept producing ribosomal proteins. The team then showed that excess copies of ribosomal proteins called RPS14 could now bind and inhibit a key protein, cyclin-dependent kinase 4, or CDK4, required to drive cell proliferation. Lessard noted immediate therapeutic implications of the team's discovery. Quote, a drug that shuts down ribosomal RNA biogenesis would immediately lead to an accumulation of ribosomal proteins outside the ribosomes. And since tumor cells make more of them, they would be preferentially affected by those kinds of drugs, he said. Added Offinger, quote, the physical interaction of RPS14 with CDK4 is the most direct link between ribosome synthesis and cell proliferation regulatory pathways discovered to date. It is therefore likely a very specific way for cancer progression to be prevented. A very specific way for cancer progression to be prevented, end quote. The study, senescence-associated ribosome biogenesis defects, contributes to cell cycle arrest through the RB pathway, was published just now, a few days ago, June 25th, in Nature Cell Biology. This is provided by the Université of Montreal. I'm proud to say that I was also a Montrealer in my youth because I'm still a Canadian citizen as well. This is on physics.org. I'll leave a link below for you for this. This is massive, massive news. Fantastic, very easy way to stop cancer. Cancer progression to be prevented in this specific way. Thank goodness, hopefully it will be implemented as fast as possible because chemotherapy is very detrimental to people taking that type of therapeutic type of solution to their cancer. because chemotherapy, of course, affects even the healthy cells of the patient.